First up will be Julie Tougas, speaking on the reproduction successions had with our galbinifrons. So, welcome. I know she's been here.
Yeah. Hi, again, smaller group. So I don't know if many of you saw my presentation that I did a couple of years ago, and it was in Tucson, Arizona. But this is kind of like almost the same thing. It's just a little bit of an update of things that have changed and some added notes on the reproduction that's occurred over the past 3 years. So for those that don't know me, I own a private breeding company in Ottawa, Canada. So I work with galbinifrons as a conservation project. And I got really lucky to have acquired my group over 10 years ago, and I love them, and they're amazing. And I'm happy to share what I've learned about them with you guys. So basically, this is what I'm going to talk about. I'm going to talk about my group, enclosure, diet, cycle nesting, egg description. I'm going to talk about my successes and failures, hatchling care and behavior, a general behavior in my group, and in conclusions. So my group, I imported them in 2002. I got really lucky. It was before all the laws changed in Canada, and I was still actually legally allowed to import turtles. That's no longer possible. And 2.3 are still alive. I had one male that died about a year and a half to 2 years later. Just suddenly, I just kind of found him dead. But the rest of the adults are doing super amazing, just as hardy, just as great as all the other box turtles that I have in my collection. When I first got them, like all Asian turtles that are wild caught, they got full treatment. So basically the Bonner treatment protocol, so antibacterial, antiprotozoal, and antiparasitic medications. Since then, I've acquired one other subadult, or more of a juvenile, that I acquired in 2012, so about 2 years ago. And I have 4 of my babies that I've managed to keep alive and raise. So I have 2 babies from 2009 and 2 babies from 2011 that I still have. So for those that were in the session this morning, you're going to see that my care methods are very different than those that were described at the Zoo Atlanta. That's basically because I'm a private collector. I work, unfortunately, in a much smaller space with less resources, but I'm very proud of what I do, and I'm obviously doing something good because it's working. My enclosure that I'm describing would be basically the minimum. I would not keep my adults in anything smaller, and my aim is to actually get them into something bigger eventually. So right now they're in an enclosure that's about 1 meter by 2 meters. I keep them on hemlock mulch. I keep all my turtles on hemlock mulch, whether they're terrestrial desert or terrestrial tropical. I'll just modify the humidity level of the mulch. This mulch is really good because it'll hold a large amount of moisture without getting moldy. They have a large water basin. I find that they spend a lot of time soaking, so I try to keep them in something that's a little bit deeper, so 2 inches, basically to almost to the top of their shell if I can. This is what I used to have as a basin, but I've actually gotten something a little bit shallow, but that's larger, so that they can get more of them all in one tub. But I would provide something bigger if I could. The lighting, so they're under, so my turtles are inside 24/7, 365 days a year. I don't have access to outdoor facilities, so all my turtles are kept under a UVB full spectrum, and I use a 5.0. And I pair it off, because I use a double fluorescent ballast, I'll just pair it off with a regular glow light or daylight bulb, just to offer the light, because the 5.0s are kind of dim compared to most other bulbs. And I offer a spot lamp seasonally. I actually haven't turned my spot lamps on at all this summer because it's been getting very hot. Actually too hot in my opinion. So my spot lamps are there, they're set up. So if I needed to get them to a thermal or body temperature in the 30s, then I'll turn it on. But I haven't needed to do that this year. And then I offer hides. They like to have their hides. If you come to my presentation later on this afternoon about enrichments, I'll talk a little bit more about hides. But I like using these little stool type things that you can get at the dollar store, and they're easy to clean when it comes to enclosure disinfection. And it's a nice height, and it's easy for them to get under and move around. The juveniles, unlike the Zoo Atlanta, I keep my juveniles aquatic. I keep all my box turtles, whether Asian or North American, in an aquatic setup for the first to 2 years. I'll move them only terrestrial when I actually see a nice shell formation. I want them to have a nice foundation structure to their shell, nice and domed, and I also want a behavior trait. I want them to be very responsive to my presence. I want them to see me, come to me like red ears. That's what I want, and that's what this aquatic enclosure does. They don't really have the option to hide and be secretive. They have to learn about my presence, and they have to learn to associate food with my presence, and then I'll move them to terrestrial. But it's more for the shell growth. It really creates a nice shell growth. I maintain the water. I have 27, 28 here, but it can drop a little bit lower occasionally, but I try to maintain it around there. And again, I'll offer a spot lamp when needed to get them in about the 30C range. And they're also under a 5.0 UVB lamp and just a regular generic daylight lamp. So diet, this is what I've been giving with a few modifications, and I think this is going to change in response to some conversations that I've had here over the past couple of days. My babies are fed 5 times a week, and I'll slowly reduce down to 2 to 3 times as they age. The babies are started on Reptistick by ZoomEd, and as soon as they're big enough, I switch them over to the natural aquatic turtle food growth formula. When they reach a year to 2 years, then I'll start seeing if they want to nibble on vegetable and fruit matter when I'm feeding it to my other turtles. And then when they become adults, I reduce their feeding to 2 to 3 times a week, and that will depend on what I gave them last time as a meal. If I give them something very calorie rich and dense like Missouri tortoise chow, I might not feed them again for a week, because that kind of just sits there and it's heavy food. If I just given them a bunch of bok choy, then I might feed them again 2 days later. So it depends on what I give them. The adults, so as protein source, I'll vary between ZoomEd, natural aquatic turtle food growth formula, and I've actually started using a dog food. I'm not a very strong promoter of using dog food in turtles, but this dog food is grain-free and it's fish-based. So it's basically fish, different types of freshwater and saltwater fish, mixed in with fruits and vegetables and with minerals and vitamins that are suitable for dogs but can't be far off from what a turtle needs. I also offer various fruits and vegetables. I try to offer it as whole as possible. I don't make salads. I don't grate it up. I try to make it so they have to work a little bit to get to their food. And Cuttlebone, 24/7 access to Cuttlebone. Juveniles, adults, any stage, they always have a piece available, and they love it. You can see them aggressively. If they ran out and there's a period before putting some in, they'll jump on it. So yearly cycle, I'll have a moist summer, drier, cooler winter. My winter season usually starts around October when I start cooling them down, and will end in March. This is something that I've been playing around with to try to increase my fertility. Let's just say this past winter I brought them down to 15, total darkness, but the substrate was a little bit damp, which is kind of not consistent with what would happen in the wild. So I might have been confusing them a bit. I'll increase the daylight around April, and I'll increase the day. So I kind of do a day cycle. I don't just keep 2 standard day cycles. I'll increase it and reduce it to try to mimic a little bit of mimic the natural light cycle that you find outside. So in the summer, I always try to maintain half the enclosure moist, and this is done just by when I change the water bowl. I'll go and dump a whole bunch of water on one side. I always offer a dry area because I want them to have that option to be able to dry out in order to prevent shell infections, plastron infections. I feel that if they're always sitting on the moist, then it's really a breeding ground for bacteria and fungus to set in their plastron. And I don't cycle the juveniles. I'll only cycle the juveniles once, in my opinion, they've hit a size where they can breed, and hasn't happened yet. So here's a nice picture of my male Simon that's trying to mount my female Blossom, and it's really interesting because you can see she's receptive. I don't know if you can see her tail. It's right there, it's sticking out. So female gal, that's receptive. It might take a little bit of convincing from the male, but if you wait, maybe 10-15 minutes later, she'll just freeze, and she becomes blank, totally blank, like she's gone in some other place, and her tail is right out, and you can see the male feels for the tail, and he uses the tail to slide over and then wraps under. So I think unless the female is in this stage, copulation doesn't occur. So the result of what we just saw turns into this. My females, what I've seen, and I've talked to other people, and their females seem to do the same thing, is a very shallow nest. In this case, she didn't even do anything. But they're quite picky. They like to have something that's a little bit sheltered, and then they'll kind of spread the mulch apart a bit, and they'll just drop their eggs, and they'll kind of just cover it, but they're not really covered. You can see them right away, and they're so big that you walk by the enclosure and they stick right out. So, and this species will just nest in their main enclosure. A lot of my other species, I'll move them into a nesting enclosure, but I just let them nest there. I reduce the group. I just keep my 3 females to reduce any damage that might occur to the eggs. And I check twice a day, and they stick right out. So like I said, the eggs are quite large. I have small hands, but they're quite large. So my eggs have been averaging 27 to 38 grams. The 38 grams come out of one of my females, huge female. My other 2 females produce more than 27 gram range, and 55 to 62 millimeters in length. Fertile eggs, in my experience, will band. They're a little bit slower to band than a lot of the other species that I work with, but I would expect them to start banding over within 14 days. If I haven't seen any whitening happening, then I'm pretty much sure that the egg is not viable. And if the egg bands, it hatches, in my experience. I've been getting 2 clutches a year, very regular. It's always the same female that lays first. It's always Rose that lays first, and then it's always Buttercup, and it's always Blossom last. Rose and Buttercup will lay 2 clutches of 1 to 2 eggs each. Blossom, it varies. She'll sometimes give me 1 and 2, or sometimes she'll just dump 3 at one time. The incubation is on moist sphagnum moss. That's also it's a consistence in my collection. I only use sphagnum moss because it retains moisture very well, and it has antifungal properties. So if you have one egg among your group that goes moldy, the mold stays on that egg and doesn't spread to the other ones. And I incubate between 27 and 30. I've been incubating for female following the Cora flavomarginata protocol. I do the first month at 30 degrees Celsius, and I drop down to 26, 27 for the second month and beyond. The hatchlings, very slow to emerge. I'm trying to remember, it's been 3 years since I've seen one, so it's quite slow. I think 2-3 days before they actually come out completely. And the incubation days duration has been between 70 and 90 days. I don't know why there's a variation between, because it's always been the same temperature for all of them. And so these are my hatch rates. And this is where it kind of gets sad. So you can see 2005 was when I got my first eggs. So these are the animals that were imported in 2002. So it took 3 years for them to acclimate. So 3 eggs the first year, I got 2 that hatched. And then 3-0, 2-2, 5-3. 2010, I got a lot of eggs. Not that good of a hatch rate. Well, it's still about 50%. 2011 was my best year for hatchlings. Out of 9 eggs, I got 5 that hatched. And then after that, dropped down to 0. I still get my eggs, stay in the same pattern. The eggs look great. Nothing is abnormal with my females. They're laying like they usually do. They're not holding them in longer. They're not giving me any complications. They're just not banding. They're just not developing. And they usually mold over within a month, these eggs. They stay in my incubator until they mold over. I don't touch an egg unless it's obviously rotting and sticky. So here's another picture of Simon again with my other female Rose. And this just occurred a couple of weeks ago. So the hatchlings, from my experience, will accept pellet food. I try to start everything on pellet food. I unfortunately don't have the time. I have a lot of turtles to actually get live involved. I'm also concerned about, introducing parasites and disease by giving live. Once they start eating, they appear to be quite hardy. And like I mentioned before, I keep them aquatic for the first 2 to 4 years. So general behavior notes. Oh, I'm actually on time. Great. I guess slow down. A general behavior note. So interspecific aggression rarely seen. So I've kept them in groups, male, females together. The only time I really see any aggression is between my males. And what was mentioned earlier is I have 2 males. One is dominant, the other one is submissive. It's clear. And it took me a while to figure this out, and it was actually the reverse of what I thought. I thought the dominant male was actually turned out to be my submissive male, or maybe they switched before I actually realized what was happening. And I've also noticed too that the dominant male, if he's in the presence of the submissive male, will spend a lot of time and energy chasing that other male out. So I'm seeing a lot more aggression between the males. And in that situation too, when I have the two males together, the dominant male does not focus on the females. He's focusing on that other male to get it out. So since then, I've only been putting my dominant male in with the females, and he goes right away, right to the females, and starts what he needs to do to get them to be in that copulatory trance so he can finish the job. My captive bred babies, they do not fit the definition of the shy, discreet galbinifrons. They are hardy, they are outgoing, they're just like any other captive bred box turtle that I own. So it's nice to see that. So the behavior of being shy is really something that the wild-caught animals have. But they do lose it, because mine were all really shy when I got them. But now, most of them are like, well, you see this picture, she's always in my face. And my dominant male Simon too, he's always in my face when I'm looking at them. I don't need to hide. They just come right up to me. They're like, food? Food? So general conclusion from working with this species, I think that I've done pretty well, except for the past 3 years of infertility. And I'm hoping that I will have found some possible solutions at this conference that I can take home and actually experiment with. Very, very long to acclimate the wild-caught animals. I've heard of them crashing several years after, and I got lucky that maybe that one male crashed, but he appeared to be a very old male. Otherwise, my turtles are not giving me any indication that they're going to crash or that there's anything wrong with them. I've had a 50% hatch rate until 2011, and then it dropped down to 0. So why? My hypotheses are incubation issues of the eggs. Not likely though, in my opinion. Improper cycling, so I've been playing around a bit with temperatures. Lack of actual copulation. I spend less time watching them right now, so I might be assuming that they're copulating, but they're not. But right now I'm really focused on nutritional. I think something might be affecting the sperm production in the males, and so that's kind of the path that I'm looking into right now, and hopefully I'm right and I can fix it. And next year I'll be back with some good news. So thank you very much. Do we have time for questions?
Yeah, a couple minutes.
Do we have any questions? Yes.
The eggs that have failed, were they fertile?
I don't know, I didn't open them up. And that's really a fault of mine. I just have a thing with rotten eggs. I gotta get over it. But I really don't think so, because fertility, I strongly believe, will be shown by banding. And they didn't band. So any egg that's ever hatched has been fertile. It's because it's the embryo that I think attaches to the egg membrane, and that's where the banding starts. So if you're not getting that cell formation, you're not going to get your banding. So that's why I just assumed that they're not good. Yeah.
What percentage of your banded eggs hatch? 100%?
Pretty much, yeah. If they band, they hatch.
You ever had any like full-term deaths?
I think I might have opened some, but those babies were highly deformed. And in my hatch rate, that's including the highly deformed, but I ended up euthanizing them. I had a couple 1 year. I don't know, it was really freaky. I had one that hatched without the plasma completely open. It was really gruesome. And I had one, I think, hatched with one eye. Yeah.
One remark and one question. Have you ever considered changing the males?
Getting new males?
No.
Or putting the submissive male? Yes, and I do that occasionally. Yeah.
Does he copulate?
I hadn't noticed. I didn't pay attention to it, no.
And the question is, have you ever considered that the females stress each other?
The females what?
Stress.
Stress each other. I don't know, because I really don't see any behavioral indication between my females that would be stressing each other.
Well, this is probably the problem. You don't see active aggression.
Yeah.
But the physical presence of a dominant female, and you told us that they have a sequence of dropping eggs. So the dominant
Yeah.
female, then the second dominant, and the last one indicate